In today's lecture at Medical Sciences, we want to look at body blood buffers, or what we call physiological buffers. These are the buffers which aid us in maintaining body pH at a constant level. So we want to see different blood buffers, whereby we are going to look at blood buffers which maintain blood pH, or what we are calling acid-base balance. And to begin with, the pH of the body. The pH of the body is always between 3.5 to 3.45 with the average of 7.4. This is for arterial blood. So this is the pH range, and below this it will result into what we call acidosis, or you can call it acidemia, and above 7.45 it results into alkalosis, or you can call it alkalemia. So our body, these blood buffers are going to make sure that the blood pH is maintained at 7.35, 7.45 with average of 7.4. That is for arterial blood. So after seeing this, we are going to see the mechanisms, how these buffers, and the blood buffers are the first line. This is the first line mechanism. Then the second line is the respiratory mechanism. The respiratory, that is the lungs. During inspiration and expiration is the second line for maintaining pH. Then the third line is the renal mechanism, whereby the renal mechanism involves the kidney. This one is the third line for maintaining acid-base balance. So when the blood pH fails, the body goes to the lungs to maintain the pH by inspiration and expiration. Then if the lung fails, then the kidney comes in to maintain. So in this session, for us, we are zeroing on the blood buffers, which is the first line. And the blood buffers we are discussing in this session, they are those buffers which are of extracellular fluid and buffers which are of the intracellular fluid, those which are found within the cells, especially red blood cells. And among the buffers of extracellular fluid, the most prominent is the bicarbonate. The bicarbonate is it is a principal buffer maintaining extracellular fluid, whereby this bicarbonate contains weak carbonic acid and a conjugate base, the bicarbonate. Then another one is the phosphate buffer. Then the last one is the protein buffer. That is for the extracellular. Then for the intracellular, still we have the phosphate buffer. We have the protein buffer. We have also the hemoglobin buffer. So these are the physiological buffers of blood that ensures the pH of blood is maintained between 7.35 to 7.45 to prevent either acidosis or alkalosis. So after looking at this, we are going to see the mechanism of action of each. And here, before we forget, the principal and the major buffer in the intracellular fluid this is the major. That is in RBCs. So we are going to begin with one by one, whereby we are going to first discuss how does the mechanism of actions of all different, the mechanism of these blood buffers, and we are beginning with bicarbonate, which we have already discussed in a certain video of the mechanism of action of buffers that is in the laboratory. So the bicarbonate buffer, briefly we are going to talk about it again, which contains the carbonic acid and a conjugate base, that is the bicarbonate. So if I have this buffer in the body, if there is anything that is triggering the strong acid on addition of a small amount of strong acid, just we are looking at its mechanism, such as hydrochloric acid. This hydrochloric acid is able to dissociate to give the hydrogen ions and the chloride ions, and these hydrogen ions formed are the ones that have ability to decrease the pH. These ones can decrease the pH to go below, be less than 7.35. So what happens? These hydrogen ions liberated from a strong acid are going to be buffered by the conjugate base, the bicarbonate, whereby the bicarbonate, that if we have the weak acid here, the bicarbonate in the solution is going to accept these hydrogen ions to form weak carbonic acid, and this acid is a weak acid, so it is dissociates partially. So we are replacing a strong acid with a weak acid, and due to replacement of a strong acid with a weak acid in the blood, it is going to keep the pH at a relatively constant level. Then these chlorides which are liberated, they are going to react with the sodium in the blood to form sodium chloride. So this is the mechanism of action of this buffer. So here the point is, how does it maintain the pH after adding hydrochloric acid? Is that the strong acid, which would have decreased the pH below 7.35, is these protons are buffered by the conjugate base, the bicarbonate, forming the weak carbonic acid, which will not alter the pH. So it will keep the pH at a relatively constant levels, because these buffers are making sure they maintain hydrogen ion concentration in the blood at a constant level. They don't remove, but they maintain at a constant level. Then another thing is, if we add small amount of a strong base, such as sodium hydroxide, this sodium hydroxide is being a strong base, it dissociates completely to give sodium ions and hydroxyl ions, and these hydroxyl ions are the ones that have ability to increase the pH. So how do we make sure that these hydrogen ions are checked or are maintained? Is that in our buffer system we have the weak carbonic acid, and this weak carbonic acid has ability to dissociate, liberate hydrogen ions and the bicarbonate, which is the conjugate base. So if there was no liberation of the hydrogen ions, these hydroxyl ions remain in the body and increase the pH, making it alkaline. But we thank God for the liberation of the hydrogen ions from the weak carbonic acid, which these hydrogen ions come and combine with the hydroxyl ion, which would have increased the pH, forming water. We form water, and this water formed is neutral, and also these bicarbonates formed come and react with the sodium, forming sodium hydrogen carbonate, which is a weak. So from this illustration, you can see the strong acid, which is sodium hydroxide, which liberates the hydroxyl ions to increase the pH, are replaced by a weak base, that is sodium hydrogen carbonate, and the water, which is neutral. Hence, these two will maintain pH at a relatively constant level. Constant level. So this is the mechanism of action of the bicarbonate buffer. After looking at this, we can also discuss another buffer, important buffer in the body, which is the phosphate buffer. Phosphate buffer. And this phosphate buffer contains the acid, and it contains the conjugate base. This is dihydrogen phosphate and monohydrogen phosphate. So this is the buffer, having a weak acid and a conjugate base. The way we were having here, a weak acid and a conjugate base. So in case in the body there is addition of a strong acid, if a strong acid is added, such as hydrochloric acid in small amounts, we told you that this hydrochloric acid, the hydrochloric acid has ability to dissociate completely to give the hydrogen ions plus the chloride ions, and these hydrogen ions liberated from a strong acid is the one that can cause decrease in pH. This one can lead to decrease in blood pH. But in our buffer, we have a proton acceptor, which is hydrogen phosphate. So we thank God for the hydrogen phosphate in our buffer, which has ability to accept these protons, and we form dihydrogen phosphate. We form dihydrogen phosphate. So at the long run, we have replaced a strong acid with a weak acid. And this one will enable us to keep the pH at a relatively constant, because weak acids do not change the pH so much. Then in case we add small amount of a strong base, such as sodium hydroxide, this sodium hydroxide will dissociate completely to give sodium ions plus the hydroxyl ions. And these hydroxyl ions liberated, they are the ones that have ability to increase the pH to alkaline if they are not removed. But remember, in our buffer system, we have an acid which can donate a proton to combine with the hydroxyl ion to form water. So this acid, which is in blood, the dihydrogen phosphate, is going to dissociate partially to liberate hydrogen ions and produce a conjugate base. So these hydroxyl ions, which would have increased the pH to alkaline, they are going to be buffered by the protons released from the dihydrogen phosphate, and we are going to have the hydrogen ions combining with the hydroxyl ions, which would have increased the pH to form water. And this water formed is neutral, so it will keep the pH at a relatively constant level. And even we had also another weak base which was produced, and this weak base will also keep the pH from rising to alkaline conditions. So this is the mechanism of action, how a phosphate buffer acts in the body. Here, when acid is added, the hydrogen ions, which would have decreased the pH, are buffered by the weak base, monohydrogen phosphate, to form a weak acid which will keep the pH at a relatively constant level. If we add a strong base, the hydroxyl ions which would have increased the pH to alkaline, they are accepted, they are buffered by the dihydrogen phosphate donating a proton, hence forming a weak base which will now keep the pH at a relatively constant level, and even the hydroxyl ions combine with the hydrogen ion to form water which is neutral, meaning it will not change the pH. That is the mechanism of action of a phosphate buffer in the body. Then another buffer we have is the protein buffer. And this protein buffer contains majorly albumin, which is responsible for buffering the action. And remember, proteins are made up of amino acids. The proteins are made up of amino acids, whereby the structure of an amino acid, this is the genome structure. Here it has the R group, then the H. So this is an amino acid. And an amino acid has a carboxyl functional group, and it also has the amine functional group, whereby the carboxyl, this one acts as the acid, acts as the weak acid, whereas this one, the amine, acts as the conjugate base. So the amine on the amino acid acts as the conjugate base, whereas by accepting proton, whereas the carboxyl group acts as the weak acid by donating a proton. So if you have such an amino acid in a buffer solution, on addition of an acid, when an acid such as hydrochloric acid is added, the hydrochloric acid gives the hydrogen ions which have ability to decrease the pH. So in the presence of the amino acid, when you add these hydrogen ions, they are going to be accepted by the conjugate base, which is the amine, and at the long run, we shall form a positively charged amino acid. So this is how it is buffered, hence forming this weak base, weak acid, which will maintain the pH at a relatively constant level. Then, in the case of addition of a strong acid, this was a strong acid. Then on addition of a strong base, the strong base like sodium hydroxide is going to liberate sodium ion and hydroxyl ions, which have ability to increase the pH to alkaline. And these hydroxyl ions are buffered. If I have an amino acid in the solution or in blood, this amino acid is going to react with the hydroxyl ion, whereby this carboxyl ion is acting as a proton donor. It donates this hydrogen ion, hence forming a negatively charged compound plus water. And the water formed is neutral, so the pH of blood will remain at a constant level. And this one becomes negatively charged. This one was positively. So that's why we normally say that amino acids or proteins in acidic media, they are positively charged, whereas proteins in alkaline media, they are negatively charged. But another point I want to bring on this buffer is that another point here to make, that you have to know that amino acids at isoelectric pH, at the isoelectric pH, they exist as Zwitter ions. They exist as Zwitter ions at isoelectric pH. How isoelectric pH is whereby pH is equal to pKa. And when the pH is equal to pKa, it exists as a Zwitter ion, whereby it is going to be having a charge of a net charge of 0. So if this is an amino acid, it has both a negative charge and a positive charge, and the net charge is 0. So a Zwitter ion is a compound with a net charge which is both positively charged and negatively charged, and the net charge is 0. So amino acids at isoelectric pH, whereby the pH is the same as the pKa, they exist as Zwitter ions. But here the mechanism of action, we have seen how they buffer an acid by forming an ammonium, and also how they buffer the base by the carboxyl group donating a proton to form water. So this is the mechanism of action of a protein buffer in the body. So we end this session by looking at the mechanism of action of hemoglobin buffer, which is our last discussion, whereby we want to look at how does hemoglobin in the red cell able to buffer the intracellular pH within the RBCs. So I want to look at in a few minutes. And if I can draw a red cell, so this is a red cell, and here we are looking at the hemoglobin buffer, which is the most intracellular fluid buffer found in red cells. So if this is an RBC, this is the tissue side, and this is the blood. So we have the cell, the tissue, and the blood. And remember, in the tissue, in the respiring tissues, they produce carbon dioxide, and this carbon dioxide can flow in the cell, a red cell. And this red cell, that is what we call this carbon dioxide in the red cell, will combine with the water, because in the cells we have water in the presence of an acid known as carbonic anhydrase to form weak carbonic acid. So here the enzyme which is catalyzing the formation is carbonic anhydrase. So carbonic anhydrase enzyme is the one that is catalyzing the formation of weak carbonic acid. And after forming this weak carbonic acid, in the presence of carbonic anhydrase, it can dissociate partially to produce hydrogen ions and the bicarbonate ions. These bicarbonate ions can diffuse into blood. It can go into blood. And remember, in blood, we have the chloride. So the chloride will shift to maintain the equilibrium of the charge. And this is what we know as chloride shift. So the chloride shifts to maintain the negative charge in the cell. After the diffusion of the bicarbonate, then this one has ability to increase the pH in the cell. But since this hydrogen ion has ability to decrease the pH, not to increase but to decrease the pH, so what happens within this cell, there is what we call hemoglobin. It has hemoglobin, and this hemoglobin has ability to combine with the hydrogen ion to form deoxyhemoglobin. It forms deoxyhemoglobin, or you can call it hemoglobinic acid. And this deoxyhemoglobin formed is a weak acid. It can easily maintain the pH at a relatively constant rate. And if this deoxyhemoglobin is taken to the lungs, it will be able to combine with oxygen to form oxyhemoglobin, And this oxyhemoglobin is the one that will enable to bring back the oxygen in the respiring tissue. Then it will release the hydrogen ions. And these hydrogen ions released, still remember we have the bicarbonate in blood. So this bicarbonate in blood can still come and react with, after acquiring oxygen, these hydrogen ions can still be buffered by the bicarbonate to form weak carbonic acid, and this reaction is occurring in blood. So this is the mechanism of action of hemoglobin buffer. We have said the hemoglobin buffer, we have carbon dioxide in the respiring tissue, enters inside the cell, combines with water in the presence of carbonic anhydrase enzyme to form weak carbonic acid. And this weak carbonic acid dissociates, bring about protons and bicarbs. And this proton and the bicarbonate has the ability to diffuse across the cell to go in blood. And when it diffuses, it is balanced by the shift of the chloride ion, which we have called chloride shift. Then the hydrogen ions which are remaining have ability to decrease the pH below 7.35, but is buffered by the hemoglobin to form hemoglobinic acid or deoxyhemoglobin, which is a weak acid, hence maintaining the pH in the red cell. However, this hemoglobinic acid can also diffuse and goes to the lungs and picks up oxygen to form oxyhemoglobin and releases the other hydrogen ion. But remember, we thank God we have already put the bicarbonate in the blood, which is able to come and combine with this hydrogen ion to form weak carbonic acid, still keeping the pH relatively constant level. So this is what we can discuss in this session. In the next video, we shall look at other mechanisms of pH regulation, which we shall look at the second line and the third line. That is respiratory mechanism and renal mechanism. Thank you so much for learning with us. In case you haven't subscribed, please, if you have understood this concept, go to the subscription button, tap there, it is free of charge, and you subscribe so that we can always notify you whenever we release such videos, so that you can continue to enjoy medical sciences. May the Lord be with you.